Hey guys, we're going to cover the first part of chapter 12 today in the Human Anatomy and Physiology book by Marieb. Um, so chapter 12 is basically, it covers the brain and the spinal cord. We're going to cover the brain in two parts. So we're actually breaking one chapter up into three parts. So let's just call this the brain part one. I actually just recorded this hour video and then I did it again without my audio recording software set up right. So it didn't record my voice. So I will do my best not to rush through it. It's a fun chapter. I think you'll like it. So we already know that the CNS is composed of the brain and spinal cord, and that these structures are contained within the axial skeleton. Beyond that, though, beyond some general gross anatomy of the brain from lab, we haven't really covered that much. And so we're going to take about 2 hours today to cover the brain in two parts. So we're going to utilize what's called the medical scheme. It breaks the brain up into four regions. So cerebral hemispheres, which compose most of the mass of the brain, about 83%. Those are wrapped around the diencephalon, which is made up of the thalamus, hypothalamus, and epithalamus. And then the diencephalon sits on top of the brainstem, which is composed of the midbrain pons and medulla. And behind the pons, we have the cerebellum. We're going to talk a bit about the cerebellum. It's an interesting structure. It used to be that we thought it was the coordination center, the proprioceptive coordination center of the brain, and it is for sure. But what we're now realizing is that it coordinates all sorts of complex activities. It's almost like an auxiliary processor, a secondary brain for complex tasks, motor and otherwise. So that's pretty cool. This last little note here, the central cavity gray matter, white matter thing, you can kind of ignore that. That's a reminder to me to mention something. I want to make sure you guys understand white matter and gray matter. So very simply, gray matter is composed of cell bodies, dendrites, synapses, and microglia. I'm sorry, not microglia, neuroglia, glial cells. White matter is composed of axons and myelination, myelinated axons or unmyelinated axons, or some mixture of the two. So white matter is just conduction transmission lines. It's like copper wires. They're axons that conduct action potentials. Gray matter, again, is where integration occurs. What does that mean? So this is a kind of a dumb analogy in full disclosure, but it works for me. Hopefully it works for you. So let's imagine you're sitting next to someone in class. It's hard to imagine now, we haven't been in class for so long, but imagine you're sitting next to someone and they're getting on your nerves. Surely you've been in that situation. I have been. Both sitting in class as a student and as a teacher, surprisingly enough. But yes, so there's been times where you get a little annoyed. So you can imagine that there is a sequence of neurons in your brain that when activated, you might just look at that person and punch them in the shoulder because you're so fed up. It's unlikely unless you have a bad temper. But you have to admit that there is a series of circumstances, and at the very least, there's a pathway that could perform that. If you chose to, you could punch that person. So let's pretend it all hinges on one neuron, which it wouldn't, of course, but let's say there's one neuron that needs to reach threshold, and you just punch him right in the shoulder. So you can imagine that there's certain things that person could do that would trigger excitatory synapses, which would bring that axon hillock closer to threshold. But then there's other things that would be inhibitory. Let me give you some examples. So maybe the person starts eating a sandwich, a bologna, dill pickle, garlic sandwich. So it's offensive on all sorts of fronts. And they're chewing with their mouth open. So it's an auditory affront. It's a olfactory affront. And it's just rude, right? So you're hearing the chomping, you're smelling the garlic dill. That's an excitatory synapse. There are neurons coming from your nose and coming from your ears, synapsing onto that cell body the one that causes you to punch the person, the one that's hardwired right to your triceps to punch them. That's not really how it works exactly, but it's an analogy. So that's excitatory. That's going to bring that axon hillock closer to threshold. Maybe it goes from -70 to -65 to -60. You're almost there. But then you think to yourself, what am I doing? I can't punch this person. I need this class for nursing school. I can't get kicked out of class. I don't want to live at home forever, or whatever your situation might be. And that's an inhibitory synapse. You have synapses coming from your prefrontal cortex, your reasoning and logic cortex, saying, whoa, 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 whoa. You got to settle down. That's inhibitory. So now you drop back down to -65, -70. But then, I don't know why I'm still dragging this analogy out. I think you get it. But there's other senses that could get involved. Emotion could get involved. They could turn to you and tell you that you look ugly. That has an emotional blow. And you might punch them in the shoulder, or maybe you say, well, no, you're ugly, right? So you can see that there's the brain is just this interplay of stimuli and then higher cognitive function like reasoning and logic. Interplaying, decisions are being made. Why did I bring this up? Oh, all of that is occurring in gray matter. All of that integration occurs in gray matter because it happens specifically in cell bodies. Okay, so you have gray matter and white matter in your brain. Generally speaking, in the central nervous system, you have a central fluid-filled cavity surrounded by gray matter, which is surrounded by white matter. And then in the brain, it's again surrounded by gray matter. So the brain's a little bit different, but that's just kind of the general orientation, this little thing I jot down here at the bottom. Oh. Okay, so speaking of that central cavity, so the center of your brain has some ventricles that are continuous with one another. So what that means is fluid can flow from one to the other. They're filled with cerebrospinal fluid, which is produced in choroid plexus, and these ventricles are lined with ependymal cells. Ependymal cells are ciliated, so this fluid is circulating within and around the brain. So you have your first and second or lateral ventricles. You have your third ventricle, which is between your thalamic hemispheres, and then you have your fourth ventricle behind your pons. This fluid is you're going to find gray matter near this fluid because gray matter needs a fluid environment for all this ion flow to occur. So it's not like blood, it's not supplying nutrition necessarily, it's supporting action potential and gradient potential formation. I'm not going to ask you much about ventricles. So here we go, we're getting to the meat here, so the cerebral hemisphere. The hemispheres form the superior part of the brain. Like I mentioned earlier, they make up 83% of brain mass. We know the lobes, so frontal, temporal, parietal, occipital, insular. I won't waste your time there. You know the gyri from lab, you know the sulci from lab, you know the fissures, so let's just keep going. Here's the cerebral cortex. This is the piece de resistance of the human body, right? This is what separates us from our animal friends. Is this amazing cortex that animals just don't have. You'll hear it in different books called the conscious mind, or an analogy, of course, not literally, but the soul of the brain, right? Where reasoning and logic and emotion all intertwine. Emotion is actually really not a part of the cortex, but anyway, it's billions of neurons, and it's 6 layers thin, so it's very thin. But despite being that thin, it makes up 40% of your brain mass because of all the different valleys and gyruses and stuff. So we're going to take the remainder of this brain part 1 video to talk about the different areas of the cortex. And it's quite fun. I have to admit, I'll be the first to tell you it's not an exact science, and we'll explain that a little bit. But anyway, there are 3 functional regions: motor, sensory, and association. This has nothing to do with what type of neuron is found there. It has to do with the function of that region. And let me explain what that means. Say I have a neuron that makes me wiggle my fingers, okay? Flex my fingers. That neuron doesn't go all the way up to my brain. It comes from my spinal cord. There's another neuron that comes from my brain that synapses on that neuron. Technically, that neuron coming from the brain doesn't touch any muscle. So it's not a motor neuron. It's an interneuron. But it's involved in motor function, and it comes from an area that is involved in motor function. So we call that area a motor area. But it's made up of interneurons. All the neurons in the brain are interneurons, to just keep it simple, even in a sensory area. So I like to ask that trick question on quizzes, what type of neurons are found in the primary motor cortex, motor neurons or interneurons? And the answer is interneurons. Because remember, motor neurons connect to muscle tissue. Sensory neurons connect to sensory receptors. None of the neurons in the brain do that. Each hemisphere, for the most part, is concerned with the contralateral side of the body. There are some exceptions, but for example, if I get a paper cut on my left index finger, I become aware of and localize that pain in the right side of my brain, specifically the right primary somatosensory cortex. Okay, so there's this crossing of the neuron tracts as they ascend the spinal cord, they cross. So, if you've ever met someone who's had a right stroke, they have left-sided hemiplegia or left-sided weakness because their stroke was on the right part of the cortex or wherever. Although symmetrical, each hemisphere specializes in different functions. Oh man, I'm going to have to burst bubbles later. How many of you are right-brained? Raise your hand. Are you raising your hand at home? Are you right-brained? Okay. There's no such thing. I know it's tough. But this whole concept of being left brain dominant or right brain dominant was actually a misinterpretation of an old research study by Reader's Digest back in like the 90s or the 80s, I don't know when. And it's stuck. It's just stuck around. So that's not to say you aren't creative, the right brainers among us. I think I might be one. Oh wait, there's no such thing. I forgot. I'm guilty of that too. Doesn't mean you're not creative or artistic or whatever. It just means that's just what you are. You are that, whether it's genetics or whatever. But one side of your brain isn't more dominant necessarily. Anyway, so sorry to bust that. It's kind of a cool thing, right? To be like, ooh, I'm a left brain or a right brain. You are whatever you still are. Don't worry. You're still good at math or you're still artistic, if that's what you are. And then, so I'm getting ready to talk for an hour, hopefully less, 30 minutes, about what the little different parts of the brain do. What this part of the brain does, what that part of the brain does. And I love this line out of your book because it's so true. Because I study fMRI. I was looking at using fMRI for a study I wanted to do for an experiment. But the thing is, fMRI is just, in a way, it's kind of flawed. And I shouldn't say fMRI. Brain research is somewhat flawed. So the disclaimer to all of this that your book uses, and I agree with, is that defining areas, so saying what an area does, is a gross oversimplification. No area of the cortex acts alone during conscious behavior. So that's not to diminish the importance of what we're about to talk about, but it's just there's a lot of gray areas. It's not black and white. Brain research is, especially functional research, is not black and white. Okay, so let's talk about the motor areas first. Now this picture has a variety of areas, some sensory, some motor. It's just a photo. But they did highlight the primary motor cortex in red. That might look familiar. That might look an awful lot like the precentral gyrus. And this is one case where the anatomical name or the anatomical area and the functional area are the same thing. So there's two cases of that. This is the first. So I could say, what is the functional name of the precentral gyrus? And you could say primary motor cortex. Anyway, so what does this area do? Well, this area initiates discrete voluntary movement. So if I decide to snap my fingers, the impulses that caused that originated in the primary motor cortex. There was some relay, so it had a synapse on another neuron to deliver the message. It's a 2-neuron chain. But the initial action potential was generated in an axon hillock in the primary motor cortex. So it initiates discrete voluntary movement. Discrete just means like, planned, specific, not like postural movement, although a lot of postural movement, some comes from here too. But, it's very specific controlled movement, I guess, and voluntary, not a reflex. So in front of it, not highlighted in this picture, but just, the region in front of the primary motor cortex, about the same size, maybe a little bit bigger, is the premotor cortex. The premotor cortex develops motor plans, a little bit more complex motor plans. So if I want to wiggle my index finger, it might not get that involved. But if I want to play the piano or write a letter, it's going to get very involved. It needs to coordinate with memory of what an A is. If I'm going to write apple, right? What's an A? What's it look like? How do I make that motor plan? If I'm going to play the piano, it needs to draw on memory of how to play the piano, what key does what, right? And it has to coordinate memory and develop a motor plan. So that's the premotor cortex. It works a lot with the cerebellum. So, that's kind of abstract. So clinically, let's think of a clinical example. Let's say we have a patient come in and he is a famous concert pianist. He plays at the Hollywood Bowl. You're working at UCLA, Ronald Reagan, and this guy comes in, VIP, he's famous, Hollywood Bowl, and he had a stroke, and now he's in recovery. And so you're going in to assess, what's going on with him. And so you ask him to, like, hey, can you touch your fingers to your thumb, and he's looking good, and wiggle your hands, and make a fist. And so he's got full voluntary and coordinated motor control of his hands. And then he's a concert pianist, so you bring in a keyboard for him, I guess. I don't know. You wouldn't really do this probably, but you bring it in and you have him play it. And he goes to play and he's like, oh my gosh, I don't know how to play the piano. Now, in real life, strokes aren't this specific. They don't affect specific areas. They're more widespread. It's an infarction that affects a whole part of the brain, so it's unlikely that he would have perfect motor control but not be able to play the piano. Anyway, my point is, as an example, let's pretend that even though he has full voluntary coordinated control of his hands, he cannot develop a motor plan to play the piano. So that would be an example of premotor cortex damaged. Can you say his primary motor cortex is damaged? Well, no, he can move his hands, but he can't play the piano. So it's not that there's a problem with a motor ability to play the piano. There's a problem with coordinating and building a motor plan. So that's the difference between the two areas. I hope that makes sense. Broca's area is just like the primary motor cortex, but it innervates muscles of the mouth and vocal cords and throat so that you can speak. It's a part of your brain that allows you to speak. There was a newscaster named Tom Brokaw. The last few classes, no one had ever heard of him. I don't think I'm that much older than you guys, although you guys keep getting younger. It seems like every year you guys are a year younger than me. So my students are getting younger. Anyway, that's a joke. I'm getting older, obviously. If you have to say something's a joke, it's not funny. My wife told me, but what does she know? So Tom Brokaw was a newscaster. He's a very unique newscaster. He had a unique voice. That's probably why he was famous. And anyway, he talked for a living, so I associate his last name, Brokaw, with this area. It's not named after him, but it helps me remember that Brokaw's area makes you talk. This is a part of your brain that makes you talk. So if you go in to a room and you say, hello, sir, can you go ahead and sit up in bed? And he does. And then you say, can you go ahead and sit on this chair? And he goes there. There's nothing wrong with his ability to understand language, right? He could understand you. But then you say, could you go ahead and tell me your name? And he shakes his head. He has to write it on a whiteboard. And then you go, ooh, I should have read his chart because now I'm being a jerk because he has Brokaw's area damage from a stroke. Okay, so you can kind of see, I think it's interesting to see what goes wrong when one of these areas is damaged. It helps you understand what they do when they're functioning correctly. Then we have the frontal eye field. Frontal eye field is, again, just like primary motor cortex or Broca's area, but it innervates or it coordinates and moves the muscles of your eye. So it allows you to look around. Okay. All right. So now let's go to our sensory areas. Our sensory areas, this is actually a list of sensory and association cortices. So sensory areas, let me give you an example of the first one, primary somatosensory cortex. If I get a paper cut on my left finger, a part of my right primary somatosensory cortex is going to be activated. And I'm going to perceive the intensity, the type, and the location of that pain. That's what it does. That is not, that's just pain. There's other examples. Touch. Okay, so I'm gonna pick up, I'm gonna grab a dime versus a quarter out of a bin. I'm gonna need to be able to feel what they feel like, but that's not enough. It's not enough to just feel what something feels like. You need to be able to associate it with memory, either from a touch sense or a visual sense. Let me give you an example. So, and we're moving on to somatosensory association cortex. It's not primarily sensory. It's associating a sense with a memory or another sense or a concept or an idea. There's some type of association occurring. So let me put that into a practical sense. So you go into a patient's room and you have them put their palm up on the table and you have like a little pen. And you say, okay, close your eyes, put both your hands on the table. I'm going to be touching different parts of your hand. I want you to tell me if you can feel it, if it's the left, if it's the right, what part of your hand is on, etc. So you touch their left index finger and they go, oh, it's my left index finger. And you touch their right palm and you go right, they say right palm. And then you push. You're like, okay, tell me if I'm pushing hard or light. And you push harder or lighter, or you have a sharper object or a duller object. And you can assess the quality of his somatosensory ability in his hand. Okay. And so say you do that and everything checks out, no problems, right? And then you say, okay, reach into this Tupperware. I have a house key. Or you don't even say that. Say, reach into this Tupperware and tell me what item is in this Tupperware with your eyes closed. And he reaches in and goes, okay, I feel it. Maybe he can say it's small, it's cold, it's metal. Maybe he can even say all those things, although he may not be, depending on how bad the damage is. But then you say, okay, what is it? And he goes, I don't know what it is. And it's like, okay, but you should know what a house key is, right? Because at some point in your life, you've felt a house key, you've seen a house key. So his association cortex is damaged because he can feel it, but he can't associate it with what it is. A memory of what a key feels like, or even what it looks like. If you have a memory of what it looks like, you might be able to associate what you're feeling with what you think a key should feel like based on what it looks like, right? So these association cortices tie that information together. They associate what you're feeling with what you know, okay? And this is developed over time. As you feel more things and see more things, the memory, you have more and more robust, higher volume of these different memories to draw on. So that's an association cortex. Each of these areas below has an association cortex, but rather than saying primary visual cortex and visual association area, I just put visual areas. Just keep it on one slide, okay? So the next one here, the visual area, you have two. You have a primary visual cortex, which is on the back tip of your brain, and then you have a visual association cortex. So you might say your visual association cortex is partly damaged. You have a patient who's partly damaged. So you go in and you say, okay, how many fingers am I holding up? And they say 2 or 5 or 3 or whatever. So they can see clearly. And they even have enough association cortex to be able to associate that with a number or know that it's a finger and all that stuff. But then you hold up a photo of a card and you say, what's this? And they're like, I don't know. Or a photo of someone they know and they don't know what that is. They don't recognize them, right? That's associating visual stimulus with visual memory. And that's what an association cortex does. Okay, if their primary visual cortex was damaged, they'd have cortical blindness. They wouldn't be able to see. So that's the difference. And so each of these has both. So let's just run through them really quick. An auditory area, that's where you perceive sound waves are converted into action potentials in the cochlea, and you interpret that as pitch and tone and volume, and you can hear things. And then you can associate that with what you know words sound like, and that's how you understand language. So say you go into a patient's room and you say, hello, sir, can you go ahead and sit up? And they don't. And then you say, what's your name? And they don't say anything. Or maybe they say cucumber spaghetti, because they don't know what words mean. But they can still talk because Brokaw's area is fine, but they don't understand what those words mean. So they can't understand you and they can't speak sensically, but they can make noise and words, if you understand the difference there. So Wernicke's area and Brokaw's area work hand in hand. I'm sorry, Wernicke's area is your auditory association area. So you can see in this picture here, there's arrows between Wernicke's and Brokaw's, and then there's also a primary auditory area, which is where you actually perceive the stimulus. Wernicke's area gives meaning to what you're hearing, and then it gives feedback to Brokaw's area so you can reply and reply in a way that makes sense. Vestibular cortex, your vestibular system perceives where you are in space, whether you're accelerating, decelerating, rising, falling, tilting. Olfactory is smell, gustatory is taste, visceral sensory is, feeling your stomach, like you have an upset stomach, or feeling your heartbeat or whatever. So these are all very simple little ones. This is where brain research is actually pretty good for simple tasks. Like, hey, snap your fingers, and then you can see what part of the brain is activated. This research is done, I don't know how much PET scan is used for this, but I know functional MRI, at least now, is used quite a bit for this. And the way it works is pretty interesting. You'll have someone sit in an MRI machine or lie down in an MRI machine, and you say, okay, we want you to snap your fingers. And what an fMRI does is it functions utilizing this response called a BOLD response. And it's an image, I think BOLD stands for blood oxygenation level dependent or something like that. And what it does is it measures what's called the hemodynamic response. So basically, if a part of your brain is stimulated, about 6 milliseconds later, there's an increase in blood flow. And because blood has iron in it, it has unique magnetic properties that show up well on an MRI. So a functional MRI is measuring increased blood flow to different parts of the brain. So all of this that you're seeing is based on that. Okay, so it's based on, hey, we're going to make a noise and see what part of your brain lights up. Or, hey, I want you to think of whatever and see what part of the brain lights up. It's fundamentally flawed, though, because your task as a researcher is to design an environment where you have all the variables controlled and you know exactly what you're stimulating, how you're stimulating that person's brain, but our brains are too complicated for that. So what we do to kind of compensate for that is we analyze hundreds of people. We have hundreds of people snap their fingers, and then we average all that data together, and we apply statistical analysis, and we say, okay, this part of the brain is most likely to be stimulated when you snap your fingers. So then we say that part of the brain is responsible for snapping your fingers. But in reality, if you look at 100 different brains, you're going to find 100 different outcomes, because you just can't control. Maybe that person's thinking about what they're going to eat for lunch while they're snapping their fingers. Or maybe they had a date last night, or maybe their stomach hurts, or maybe they're mad at their landlord, right? It's just difficult to really study the brain. But if you get enough people together, that background noise should kind of average and cancel each other out, and you're left with the strongest stimulus, which is when you're snapping your fingers. That should be the dominant area that's stimulated. So if you get enough subjects and do a statistical analysis, you can map the brain, and that's how this stuff is done. But again, it's not an exact science, to say the least. So, okay, 2 more slides, and I think that will wrap up part 1 of part 1. So it's like part 1A. This is the brain part 1. So we'll do a brain part 2 this week, and then next week we'll do a spinal cord. That's all chapter 12. So we're breaking chapter 12 up into 3 parts. We're going to do 2 this week, 1 next week. So anyway, multimodal association areas. I don't know if you guys are familiar with the term modality, but it just means a different type of something. So if we're talking about sensation, you have vision, you have hearing, you have pain, you have temperature. Those are all different modalities of sense. So a multimodal association area is able to tie multiple modalities together and coordinate with other areas of the brain. This is a higher level of brain function and therefore even more difficult to research. So your book gives a good example. I'll read it. So it says, suppose, for example, you drop a bottle of acid in the chemistry lab and it splashes on you. You see the bottle shatter, you hear it shatter, you feel your skin burning, you smell the acid fumes. Man, that's a lot of different stuff. We have a visual stimulus, we have a auditory stimulus, and pain from burning, and smell from the acid. You probably also have a feeling of danger that comes from somewhere else, which we'll talk about in a second. Anyway, all these perceptions come together in the multimodal association areas. Along with feelings of panic, these perceptions are woven into a seamless whole, which hopefully recalls instructions about what to do in this situation. As a result, your premotor and primary motor cortices direct your legs to propel you to the safety shower. So we can break these multimodal association areas into 3 general multimodal association areas. So the first is the anterior association area, often called the prefrontal cortex. This is the most complicated cortical region of all, and it's involved with intellect, complex learning abilities, recall, personality, working memory, abstract ideas, judgment, reasoning, persistence, planning. It's the most slowly developing area of the brain, which, although kind of is a bummer because I have a 3-year-old who's a maniac, right? Sometimes I wish she had a fully developed prefrontal cortex and she would just be, a little adult that I didn't have to. No, not really. I love out. 3 is a good age. I disagree with the terrible 3 idea. But anyway, it's a good thing because you want the development of this area in particular to be based on a lot of data. You don't want to lock them in as a fully functional adult at 3 years old because they only have 3 years of experience to form their personality, and that's just not enough. So this takes much longer to finish developing. I think you're like in your mid-20s when this is more or less done developing. You're always developing. I mean, your brain pathways are always changing, but it's more or less complete. Anyway, posterior association area. So the posterior association area is very large. It encompasses parts of the temporal, parietal, and occipital lobes. It helps you recognize patterns, faces. It localizes you and your surroundings in space. So you know, okay, hey, my teacher's way over there, but oh man, the girl I like is right over there and she just saw me drop acid on myself. That sucks, right? you're figuring out where you are, where objects are, the safety shower's over there, and where people are. Although that emotional context actually comes from somewhere else, so that the girl you like may have been a bad example. Anyway, your awareness of the entire scene originates from this area. And it plays a big role in written and spoken language because it encompasses Wernicke's area. And then lastly, the limbic association area. It's a part of the limbic system, so it includes cingulate, parahippocampal gyrus, hippocampus, and it provides the emotional context and impact that makes something important to you. So in the example above, it's providing that sense of danger or perhaps embarrassment, depending on the situation. Whatever the emotion may be, that comes from the limbic association area. And then lastly for this part, and then we'll move on, is lateralization of cortical functioning. So this comes back to that left brain, right brain idea. When you look at this table, you get the idea that you can be left cerebral dominant or right cerebral dominant. And I put it in here on purpose because whenever this study came out that showed that different parts of the brain specialize in different things, they made a mistake, I think, personally. They didn't make a mistake. The reporter that read the article and misreported it made the mistake, but I think they chose their words poorly. So basically what they said is, ignore this bottom part for a bit right here. Okay, just ignore that for a second. They made this table, okay, and they said, in most people, I don't know how many people, but this is how the brain is structured. Most language comprehension and production centers are on the left. Math centers, logic centers are on the left. And the right brain is more involved with emotional things and artistry and music and facial recognition, that sort of thing, in most people. And then they were like, well, it's not really most people. This is how it is if you're right-handed. Most people are right-handed, which is why most people look like this. And so what they said, okay, if you're right-handed, then let's say you're left cerebral dominant, because that's where language, math, and logic are. So we'll call that the dominant side. And we'll call art and emotion, that's not the dominant side. So they say right-handed people have left cerebral dominance. So, and then left-handed people, we just need to reverse this. So the left hemisphere is where, a lot of emotional cognition takes place, artistry, music, that kind of thing. And the right side is where math, logic, and language is. That's if you're left-handed. So they said, we'll call left-handed people right cerebral dominant. So that way, instead of writing right-handed versus left-handed, they can write left cerebral dominance versus right cerebral dominance in their paper that they were writing. So someone read this who worked for Reader's Digest and said, ooh, if you're left brain dominant, you're better at language, math, and logic. It's a subtle difference, right? But you can see how it's completely gotten out of control, and now we think some people are right brain or left brain. That's just not the case. Some people are just better at being creative, and some people are just better at math. It's not a cerebral dominance issue. Your cerebral dominance is based on whether or not you're left-handed or right-handed. Okay? So that whole idea is called lateralization of cortical functioning, meaning that certain tasks are performed in certain parts of your brain. That's all it means. In 10% of people, there's no clear division of labor in this sense, right? Those people are ambidextrous. They're left and right-handed. So there's a very strong connection between your hands and your brain. That's why writing things or drawing an anatomy or coloring and labeling notes is so important. Sometimes it seems silly, but using your hands somehow, if you can, while you learn, and really typically that's in the form of note-taking, that's the only way, or drawing or sketching, is huge for learning. So anyway, okay, so I think that's it for part 1A, the brain part 1. I'm going to post these both in one course. So this week your lecture will be titled The Brain, and next week it will be titled the spinal cord. So you don't have to wait forever for this lecture. I'm going to go ahead and post part 1 now, and I'll probably post part 2 tomorrow. Okay, guys? I'll see you in the next video. Thanks.